Hello students, welcome to my botany class, and today we'll discuss a very important and essential part of the chapter cell cycle, which we started in another video lecture. Today we'll continue with the meiosis. Now meiosis, as you know, is part of sexual reproduction in organisms. It's not just syngamy or fertilization alone, but meiosis also plays a very important role in sexual reproduction. Why? Because meiosis is not only a method of cell division, but also meiosis brings about a variation, and this variation is by means of a process called crossing over. So we will discuss all the nuances, all the details of this process called meiosis. Now, before we start with the process, you need to know that meiosis is 3 types. I've written here, and also where we study it, I've written here that it is in onion anther. Now, that is a very common material used even in the school practicals and all. Though it can be in any other anther also. Wherever there's a pollen tetrad formation from the pollen mother cell, there's a process of meiosis, and it's very easy to study. Though you can use animal also. At the time of gametogenesis in animals, you know that there is a meiosis process in higher animals. Now, these are the 3 types, that is the gametic, zygotic and sporic. So why these are 3 types of meiosis? Because there are different kinds of organisms on earth. Now, if the organism is itself a diploid, then there is a gametic meiosis, because diploid organisms, they form their gametes by means of meiosis. Meiosis does what? Meiosis does not only a cell division, but it also reduces the chromosome number to half. Now, that is the most important part of the meiosis, reduction, division, which we'll study. So in that, the chromosomes, suppose human beings 46 in the diploid somatic cells, will get reduced to 23 haploid n number in the gametes. So this is achieved through meiosis, and it is essential part of the alternation of generation and sexual reproduction, because the 23 male and 23 female, these gametes, when they fuse, they again restore the number 46. So this is what happens in all the diploid organisms, and this is essentially a gametic meiosis, and we call the life cycle as diplontic. It is not only in the higher animals, but many other, even the plants and all. But in plants, there is one thing that we may not call this as gametic, because in plants, the spore formation, which is reproduction purpose, sexual reproduction purpose, the microspore, which is pollen, and the megaspore for the female gametophyte formation. During that time, there is a meiosis in the spore mother cells. So you call in plants, higher plants I'm talking about, this meiosis as a sporic. So I've written a sporic. Now, if higher animals have a gametic and the higher plants have a sporic meiosis, then what is the zygotic meiosis? Now, zygotic meiosis happens in some lower organisms, especially the fungi, then many algae, where you find the organism itself is haploid. Now, when the organism is haploid, there is no question of meiosis at the time of gamete formation. So what they have is a zygotic meiosis, because the zygote becomes diploid, and there it is a requirement of zygote to get this meiosis done, so that the number gets haploid again for the somatic cell formation. So that is how this process is known as zygotic meiosis. So 3 types of meiosis we have discussed. Now let us proceed with the meiosis itself. In meiosis, there is, as usual, this is also cell division, so no cell division can proceed without interphase. Whether it is a mitotic or meiotic, the interphase is a must. So mitotic, we have discussed in another video lecture, and before that I have discussed the interphase as well. So interphase is common, it is same with the same similar checkpoints. So in interphase, there is a G1, there is S and G2 phases. So that part remains the same, will not go into that. Coming to the M phase of meiosis, the M phase here consists of a karyokinesis and of course followed by a cytokinesis. Now the karyokinesis in meiosis is of 2 types. It's not 2 types only, it is actually 2. Nuclear division is twice. In meiosis, the nucleus divides twice. Now, when the nucleus divides twice, it means there is a meiosis 1, which we call as a reduction division. The meiosis 1 itself is called a reduction division. And in this, we will see that this is the basis where we find that the chromosome number gets half, actually. We'll see how it gets halved. Now, beginning with the stages, you remember already mitotic karyokinesis that the stages were prophase, metaphase, anaphase and telophase. So those stages remain the same, but only thing is that here the prophase 1 is pretty long, it's quite long because of very complex phenomena which are taking place in the prophase 1. Now this is the longest and the most complex I've already written here. You can take a snapshot of the entire thing that I have given here. Okay, and then we can proceed that here. To begin with, the prophase 1 has 5 stages. You can see these 5 stages: Leptotene, Zygotene, Pachytene, Diplotene and Diakinesis. So these are all given here with diagrams. Now you can see the first, that is Leptotene. Now what happens in the Leptotene? To begin with that, I have written here that I am taking here, considering a cell which is 2n, diploid is equal to 2. A parent cell which is diploid, 2n is equal to 2. Now 2 means that it is one pair of homologous chromosome, one maternal, one paternal. These things we have discussed already earlier. So this is a, you can see it's a homologous pair of chromosome. I am showing one with black and the other with a blue pen. And the DNA, it has already doubled, as you know, in the interphase S phase. The DNA doubles itself. It's a synthetic replication phase from 2C. Suppose the parent cell had 2C. So the DNA has already become 4C. So I have written here 2C has become 4C. So this cell is 4C right now. Now Leptotene is a very early prophase 1. So very thin thread-like, you will see the chromatin matter. I have though made it a little thicker so that you can see, but actually it would be much thread-like. Now all the telomeric ends of the chromosomes will remain attached to the nuclear membrane, to the nuclear envelope, and it's near to the centrosome. As I'm drawing here, it is near to the centrosome. Now since here is only one pair I'm considering, so it appears as if they have paired. No such pairing. Remember that in Leptotene there is no pairing of the homologous chromosomes. They just remain randomly and they just form a kind of bouquet or basket. So I've written this as a bouquet stage. All the chromosomes, they remain as if they remain like a basket or bouquet. So this is a bouquet stage, and once you go from here to the next stage, that is zygotene. The second is a zygotene. Zygotene, a very important stage where firstly you can see what are the changes here. You can see that the centrosome, it moves from one end to the other. So that means they are moving to the poles. The centrosomes were here, lying here, as you can see in Leptotene, but now they're moving from one end to the other. So they move to the two poles. And secondly, the homologous chromosomes, they start pairing. Now the homologous chromosomes start pairing something like this randomly. If there are 46 chromosomes, so you understand that each chromosome 1 has a pair, 2 has a pair, and these all come up together. So these pairs each having 2 chromatids, because that you already know that it is a post interphase, post S phase. So all the chromosomes, the DNA has doubled. So instead of 1 chromatid, they all are 2 stranded, 2 chromatids. So you can see this, these are 2 chromatids. So homologous pairing has happened in zygotene. Not only that, this pair also is getting close and close, and they actually form a complex. They come together and they form a complex with the help of what is known as the nucleoprotein complex, which I have written here. The phenomena is called synapsis, and the complex is called a synaptinymal complex. The complex is named as synaptinymal complex. So this complex is a nucleoprotein complex which joins, which makes it join. The bivalents come together. If I try to show you the bivalent, it's a bivalent. You can mark this word. This is called a bivalent. Why it's a bivalent? Because just one chromosome which has its own homologue, and these maternal paternal, they have formed a pair. They are inseparable right now, they are joined, so this is called a bivalent, and it has happened with all the 23. So therefore it is 23 pairs right now. If you see, there will be 23 such bivalents. Okay, those 23 pairs have all formed how many bivalents? They have formed 23 bivalents, each having 2 chromosomes. So that makes it the total chromosome 46. But bivalents are how many? 23. Okay. Now I was trying to tell you. Now if to begin with we had a homologous chromosome, this is one, and say paternal, and this is another which is maternal, and this was a homologous pair of chromosome. Now since after S phase and by the time you enter prophase 1 zygotene, they all have now these are 2 chromatids for each. Each chromosome has 2 chromatids, and these are well visible right now. Now since these are well visible, so what you see is something like this. So one chromosome and it's another homologue, that is a pair, they are like this. Okay. Now these 2 chromatids of one chromosome, they are joined by their own centromere, and these 2 chromatids of the other, say this is paternal, then this is maternal, this is also joined by its own centromere, if you can make out. So this what I draw here is nothing but the centromere. So if you want, I can write it somewhere, maybe here. So this is centromere, if it is visible in this diagram. The same is here, and each centromere has 2 kinetochores, which I have written here in the next stage, that is Pachytene. Each centromere has 2 kinetochores. Okay. Now if you have understood till here, this synapsis process, which is also even called as a syndesis, the synapsis is even called as syndesis. Then we can move on to Pachytene, a very important stage, that is Pachytene. Now what happens in Pachytene is that now the proper tetrads are visible. You may call this as bivalent only till here in zygotene. But bivalent is also a tetrad, because you know that each has 2 chromatids. Each chromosome has 2 chromatids. But it's visible right now in Pachytene onwards. Now in Pachytene, you see there is a centromere, and there are 2 kinetochores for each. So that makes one bivalent. How many chromatids? 4. One bivalent, how many chromatids? 4. Okay. And how many centromeres? 2, because each has 1. So 2 centromeres. How many kinetochores? Each centromere has 2. So therefore 4 kinetochore. So this part should be very clear in your mind. If you want, I can repeat that a bivalent consists of 4 chromatids. So it's a tetrad. It has one centromere each. So there is 2 centromeres. It has 2 kinetochores in each chromosome, so there are 4 kinetochores. So accordingly, if it is 46 chromosome, or if it is any number given in your questions, you have to quickly calculate. There is 46 chromosomes, and how many bivalents? 23. How many centromeres? Obviously 46, because bivalent are 2 chromosomes, that means 2 centromeres. So 46. But how many are the kinetochores? Of course, the kinetochores are going to be how many? 46 into 2. Or that is same as saying 23 into 4. Okay. So coming to Pachytene, crossing over. Now the most highlighting feature of the Pachytene is crossing over. Crossing over is a phenomena where the non-sister chromatids of 2 homologous pair of chromosomes, they engage themselves in what is known as crossing over, that is a breakage, exchange and reunion of the parts, not the entire chromosome, of the parts. I'll try to show you with the pens that what is this crossing over. It is a breakage, exchange and reunion of parts of non-sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosome, what does it mean? Now if you look at this, that this bivalent, this is one chromosome I have written as the 2 chromatids 1 and 2. This is another chromosome, it's homologue, and I've written the chromatids as 3 and 4. Now remember, 1 and 2 would be the sister, 3 and 4 are going to be the sister chromatids. Then what are the non-sister chromatids? Of course, any combination, it may be 1 and 3, it can be 1 and 4, 2 and 3, or 2 and 4. So these all are non-sister chromatid options. Depending on how these are, intertwined and all, it can be a crossing over possibility between any non-sister chromatid. Now, I am showing a crossing over, say, between 2 and 3. So can you see that I make in this black, I make some blue here, some blue here. In this blue, I make some black here and some black here. Why? Because this got exchanged. It not only broke off, but they got exchanged with the help of enzyme called recombinase. And these points, maybe this point and this point where the crossing over has occurred, you may call them as recombination nodule. So this is a recombination nodule. Enzyme which has helped is a recombinase enzyme. Okay, and this would be a source of variation. See, since in genetics further we also discuss about how the genes get influenced, how the recombination takes place, etc. But firstly, now for the time being, you can understand that it's something like when your parents, in your parents' body, when the gametes were being formed, at that time, your grandfather and grandmother, both sides, that is grandfather, grandmother of your father as well as of your mother, they are exchanging a dialogue, because in your parents, the maternal and the paternal set of chromosomes, they are of course one from your grandfather, one from your grandmother. So it's something like they are talking, they are exchanging and talking how to design you, the next generation. Parents are only the instrument, and these maternal paternal chromosomes of your parents and the testes and ovaries of parents, they are kind of talking. It's a dialogue that how to design you, that is the next generation. So this is very interesting, and you can see this is the homologous pairs of chromosome, and this is already a bivalent or tetrad I explained just now. So you can see, if you look at the combination here, the pens, look carefully, blue with blue and blue with blue. So this is one. Say this is maternal. Black with black, black with black, this is the paternal. Now what I'm saying, that these two are sister and these two are sisters. So non-sister chromatids can exchange. Now if I say that this blue and this black, they exchange. Now when they exchange, they undergo the crossing over, I pick up these lids and put them here. I do exchange and do like this. Okay, now this crossing over has occurred. Look at one thing, that all the genes which might be present here, no longer they are in the same combination with this part. The genes of this are different now with this part. Why? Earlier it was something like, blue with blue, but now you can see it is something, whatever you call it, black. I was calling it black. So black with blue. Similarly, in this pen, it was a black with black, but the combination has changed because this blue has gone here. So this is the blue with black. Now this is how the gene shufflings take place. Whereas this is unaffected pen and this unaffected pen, which are chromatids, you will call them as non-crossover chromatids. So there is no change in these. And these two, where recombination has occurred, you call these two as crossover chromatids. So this is a very essential part of what is known as crossing over, variation, etc. and genetics. When you study especially, you will understand that how important this is. Now going from here, I think Pachytene is clear to all of you. So, and one more thing, you can see that these centromeres have almost reached the opposite poles by now. The fourth stage of the Prophase 1, which is called as a Diplotene. In Diplotene, there's a very important thing that occurs, that is called chiasmata formation. Now what is chiasmata, or X-shaped bodies, as I have written? And this is a bit low power view that I am showing here. This is high power view that you are seeing. Now low power view, I am showing that these crossover points, which were the recombination nodules, are still sticking to each other, which means the crossing over is not yet over. It is still continuing in Diplotene. But the X-shaped bodies are where it is continuing. But why they are X-shaped visible now? Here the whole length was fused, nothing was X-shaped here. So why it's visible now? Because the non-crossing over points have separated. That means the synaptonemal complex has separated or dissolved in the non-crossing over points. As a result, you see this X-shaped bodies. Now these X-shaped bodies or chiasmata, okay, are the highlighting feature of Diploteam. Moving on from here, from Diplotene to Diakinesis. Now in Diakinesis, you can see that it is a terminalization of chiasmata. Okay, what does it mean? Diakinesis is a terminalization of the chiasmata. The chiasmata points are moving to the terminal ends of the chromosomes. And in between you find a wider gap. So this is a wider gap and the chiasmata points are moving to the terminal ends of the chromosome. It's still a bivalent, no change. It's still a bivalent. So don't get confused. Everything is same in terms of chromosome number and the DNA content. Going to the next, that is, in Diakinesis also, since it is coming to an end of the prophase 1, you need to remember that by this time, the nuclear envelope nucleus will disappear, the spindle formation will begin, and the chromosomes are quite condensed by now. As you know, condensation began from zygotene onwards, a little bit here also in Leptotene, but zygotene onwards, and it will continue till metaphase, the condensation of chromosome. Metaphase 1, a very important part of the next, that is after prophase, part of the meiosis 1, metaphase 1. By this time you can see that the centrosomes move to the opposite poles, have already formed the aster, and the microtubules of the aster orient or help in the formation of these spindle fibers. So spindle fibers are formed in metaphase 1. You also know that asters are formed only in the animal cells and not in the plant cells. So in plants we call it anastral. As in mitosis, same in meiosis, it is anastral and it is amphiastral. Amphi. The word amphi is used for the, both two. So two astrals are there. So we call the animal meiosis and mitosis as amphiastral. Now this spindle is formed, it's okay, and what you see, there is a congregation. There is a congregation, means coming together. Congregation of all the homologous pairs of chromosomes, they all arrange at the central equatorial plate, which you saw even in mitosis. But only thing is that these are two, the homologous pairs, since there are two chromosomes, so each bivalent, when they arrange, we see this as double metaphasic plate or double equatorial plate, because there are two centromeres for each bivalent. So I draw it with a dotted line. You can see this is one metaphasic plate and this is another metaphasic plate corresponding to two centromeres. There are two kinetochores each. So only thing is the spindle fibers which attach to the kinetochores would be with one each, one chromosome connecting with this centromere and the other chromosome connecting with this centromere, unlike in mitosis where both the kinetochores were attaching with the spindle fibers. But for bivalence, in the metaphase 1 of meiosis, only one kinetochore of each will connect to the chromosomal fiber, spindle fiber. So this is 2n is equal to 2, the cell remains same, chromosome, and even the DNA content 4C, no change. Going to the next. Now you see there is Anaphase 1. If you can see the Anaphase 1 diagram, in Anaphase 1, this is 2n is equal to 2 for sure. I've written, and it is 4C, and you may think that whereas in mitosis it was anaphasic doubling of a chromosome because the centromeres would divide. But in Anaphase 1 of meiosis 1, there is no such centromeric division and the chromatids do not separate. But what separates? Till now it was a bivalent. So right now the univalents will separate. That means one chromosome with its two chromatids, another chromosome with its two chromatids. Univalents will separate. They are even called as dyads. So I've written here, these are univalents or dyad separation. Univalent or dyad separation. And you can see crossovers already happened. So whichever chromosome, even if you see NCRE or any other book, you will see whichever chromosome you are concentrating, you can see that they have undergone crossing over, and the combination of the two colors you can still see here and here, like that. Why? Because this is meiosis 1. So univalents have separated, chromosome number still remains same. You can't call this as 4 because the centromere has not divided and they are still sticking, the two chromatids are still sticking to the same chromosome. So chromosome number remains the same. Next, telophase 1. Telophase 1, even for mitosis you already have studied. it is a kind of stage where the two nuclei are forming back. So in one cell now there's a formation of two nuclei, and these two nuclei, since they are getting formed, so in the cell the situation remains 2n is equal to 2 and 4c same. But yes, in this cell the two nuclei, where you see one univalent each, this univalent in this one, this univalent gone into this one, nuclear envelope, nucleolus, Golgi body, etc., etc., they all come back, they reappear. This part you already know. So that means if you concentrate on one nucleus, it is n equal to 1 and the DNA content 2c, n equal to 1, DNA content 2c. So that means it's already the reduction done. So why this meiosis 1 is called as a reduction division? Because you can see by this time the nuclei are already having one univalent. That means out of the paternal, where the dialogue took place, where the exchanges took place, and right now it is only one chromosome, either maternal or paternal, has moved into each nucleus. So therefore the nucleus is carrying n equal to 1. We can't call it 2n, n equal to 1, because it's only one of the homologue, and the DNA content is 2c. But if there is a cytokinesis right after this, then there will be two cells formed with n1 condition, or it can be first a mitosis like meiosis 2. After meiosis 1, there can be a meiosis 2, and then only there is a cytokinesis. I mean, there are two kinds of cytokinesis possible, either after telophase 1 and then again after telophase 2. This kind is called as a successive cytokinesis. But if it is a telophase 1 followed by meiosis 2, which is like mitosis, and then thereafter there is a cytokinesis where at a time 4 cells are formed, you call that as simultaneous cytokinesis. So whichever it is, but ultimately we know there are tetrad to be formed. There will be 4 cells. Product of meiosis would always be 4, tetrad. Meiosis 2, I have not drawn any diagram because this is equational division just like mitosis. And only thing that you need to understand is that it will continue with the same nucleus, same cell or same nucleus, whichever, depending on the cytokinesis. So it is having n equal to 1, and mitosis is like a photocopy, we have already discussed. And in this mitosis, there won't be any interphase because interphase is only once and it has already occurred before meiosis 1. So that means it is n equal to 1, which is going to be divided into 2 cells, copied. It's just like a copied. So it is going to be n equal to 1, 2 cells. Similarly, this n is equal to 1, 2 cells. That means altogether there will be 4 cells having n equal to 1 chromosome, and DNA content will be 2c in, DNA content will be cc in each. Once it divides, here it is 2c, it's okay, but after meiosis 2, n equal to 1, because these chromatids will separate in Anaphase 2. I hope you understand. The chromatids will separate in Anaphase 2, and momentarily the chromosome number will double up. That you have to take care. Chromosome number in Anaphase 2 will double up. It will become n equal to 2 for the time being in each cell or each nucleus. But at the end, it will be n equal to 1, 4 cells, and it will be CCCC. The DNA content I started with was 2C. So the parent cell, the DNA content was 2C. I've written here. So it became double, so 4C. But by the time it is distributed in the 4 daughter cells after meiosis 2, it becomes CCCC. Each cell having 1 chromatid. As per this diagram, each cell will have 1 chromatid, but it will be called as n equal to 1 chromosome, so n equal to 1, and the content will be C, so it is CCCC. So 4 such daughter cells will be formed, and you call it as a tetrad. So coming to the end of this, and one term that we have missed here, that is interkinesis. Interkinesis is the gap between telophase 1 and meiosis 2. So it is the end of telophase 1 and beginning of meiosis 2, so called as interkinesis. So with this we come to the end of this very important topic of cell cycle called as meiosis, and it's a basis for sexual reproduction and variation. So follow this, go through this, repeat the video again and again to understand, make notes and enjoy. Bye for now.